everyone, welcome to TryRide. This is Pack 365, myself Srija. So today in this session you are going to learn about immune cells. So what are immune cells? So immune cells, these are called as white blood cells or WBCs or also leukocytes. So these are the protective soldiers of our body. So their main job is to defend us from anything harmful such as bacteria, viruses, fungi, parasites, toxins, cancer cells, any foreign substances. So think of them as an army where each soldier has a specific role but they work together to keep the body safe. So what are the main functions of immune cells? So they recognize and destroy pathogens. So they can detect the non-self organisms or harmful substances and attack them. They produces antibodies. So some immune cells, mainly B cells, they make antibodies which are special proteins that lock onto germs and mark them for destruction. So it also kill infected or cancer cells. So cells like T cells and natural killer cells, they can directly kill the virus infected cells as well as the cancer cells. So they act as like assassin cells and they present antigens to active other immune cells. So these cells such as dendritic cells and macrophages, they show the pieces of germs, antigens to T cells. So this is like saying, look, this is the enemy, get ready to fight. It also produces the chemical messages, the role of cytokines. So cytokines, these are the signals that help the immune cells to communicate, to call for backup, to increase the inflammation and to control the healing. So you have to think them as like a walkie-talkies for the immune cells. So immune cells, they also develop the memory. So some cells become memory cells after an infection. So they help the immune system respond faster and stronger if the same germ attacks again. So this is the basis of the vaccination. So immune cells, they also maintain the inflammation and healing. So certain immune cells, they help the start inflammation to fight infection. They stop inflammation when healing starts and they repair the tissues. So this can also clean up or the dead or damaged tissue like cells like macrophages. They act as a cleaning crew. So they remove the dead cells, damaged tissues, debris after infection. So without immune cells, the body cannot fight infection, heal wounds or protect itself. So what are the types of the immune cells? Here there is a picture of the cells of the types of the immune system: T cell, B cell, natural killer cell, monocyte, macrophages, neutrophils, eosinophils, basophils, mast cells and dendritic cells. So let's go into brief description about these cells of the immune system. So classification of the immune cells. Now how the immune cells are divided. So they are divided into two main groups. The one is innate immune cells, the other is adaptive immune cells. Now what are innate immune cells? So these have fast, non-specific and there will be no memory. So these innate immune cells, these are the cells which are the body's first line of defense. So they are ready to act immediately when a germ enters the body. So they do not need training or previous exposure. So what are the key characteristics of this innate immunity? So fast, nothing but they act within minutes to hours. It is non-specific, like it attacks any foreign invader. It has no memory, right? It responds the same way every time. And these innate immune cells, they are present from birth. So what are the types of this innate immune cells? Neutrophils, eosinophils, basophils, mast cells, monocytes or macrophages, dendritic cells and natural killer cells. So these are under innate immune cells which are present from the birth. So next is adaptive immune cells. So these adaptive immune cells, they are slow, specific and they have the memory. So the system is like a trained army. It takes time to activate but it is very precise and it remembers all the infections. Now what are the key characteristics of this adaptive immunity? It starts very slow. Like it takes long days to develop during the first infection. And these are highly specific, like they targets a particular germ and it has a memory, so it responds faster next time. So it also improves over time, right? So what are the adaptive immune cells? B lymphocytes, nothing but B cells, plasma cells, T lymphocytes, and within T lymphocytes we have helper T cells, cytotoxic T cells and memory T cells. So the first type is neutrophils. So neutrophils, these are also called as polymorphonuclear leukocytes. So neutrophils, these are the most abundant white blood cells and they act as the first line of defense against the infections, especially bacterial infections. So they are very fast, aggressive and also short-lived like emergency soldiers. Now what is the structure of neutrophil? The size of neutrophil is a rounded shape. So these are the round cells which varies about 10 to 12 diameter, about the same size as the red blood cell but slightly larger. So it has a multi-lobe nucleus which has 3 to 5 lobes. So the nucleus looks segmented and these are broken down into 3 to 5 parts. So this is why they are called as polymorphonuclear. The meaning of polymorphonuclear is many lobe nucleus. So it also has cytoplasm with fine granules. So the cytoplasm contains tiny granules which are filled with chemicals that can kill the bacteria. So these granules, they contain myeloperoxidase. This creates the bleach-like substances to kill the microbes. It has defensins which punch holes in the bacteria, and this contains proteases which breaks down the proteins of the microbes. So these are like chemical weapons which are stored inside the cell. So now what is the origin of neutrophils? Like where they come from? They are produced within the bone marrow from myeloid lineage. So these are released into a blood in a fully mature form. They live for about 6 to 8 hours in blood and 2 to 3 days in the tissues. So they die after fighting. So most pus is made up of dead neutrophils. So now what are the functions of the neutrophils? What does this neutrophils do? So they act as the first line of defense against the bacteria. They are the fastest responders whenever bacteria enter the body. So second function is phagocytosis. So these neutrophils, they eat and swallow microbes into a sac called as phagosome. So they produce the reactive oxygen species. So neutrophils, they generate powerful chemicals like superoxide, hydrogen peroxide to kill the bacteria. So this is called the respiratory burst. And they also releases the enzymes. These enzymes, they break apart the microbes outside the cell. So they form the NETs. So they form the neutrophil extracellular trap. So when neutrophils die, they release the DNA and proteins as a sticky web. So these webs, they trap and kill the bacteria. So here is a diagram of the neutrophil. So this neutrophil, what does it contain? It contains the multi-lobe nucleus, right? You can see clearly in this picture. It has multi-lobe nucleus, a central dark purple structure. So this shows 3 to 5 lobes typical of neutrophils. And next is granules. So small colored dots which are scattered in the cytoplasm. So these granules represent the enzymes and antimicrobial chemicals. And next, phagosome. So phagosome is a bubble-like structure inside the cell. So this is where the bacteria are trapped and digested. And here it is glycogen. Glycogen are nothing but this is stored energy. So stored energy granules. So glycogen, it helps the neutrophils to survive in low oxygen areas like infection sites. And it has Golgi apparatus which helps to package the enzymes into granules. It also has the lipids which were stored fats used for energy and membrane structure. And it has mitochondria which provides energy. So neutrophils, they rely more on glycolysis than the mitochondria. So works even in low oxygen. So next is the last part is cytoplasm. So this is a fluid part of the cell which is containing the granules and organelles. So this is about the neutrophils. So next cell is eosinophil. So what are eosinophils? These are white blood cells mainly involved in the fighting parasites and allergic reactions. So they have a very distinctive appearance that make them easy to identify in the blood smears. So what is the structure of the eosinophil? Bilobe nucleus. So the nucleus has 2 connected lobes resembling a pair of spectacles or glasses. So this is one of the identifying feature. And it has red or orange granules in the system or in the cytoplasm. So the granules stain bright red or orange with the acidic dye eosin. So this is why they are called as eosinophils. So these granules, they contain the toxic proteins that kill the parasites. So what are the granule contains? Major basic protein MBP and eosinophil peroxidase. So major basic protein, it is the main toxic protein that damages the outer surface of the parasites like worms or helminths. So it has this eosinophil peroxidase, like it produces toxic radicals to destroy the parasites. So you have to think of these eosinophils as a parasite killing cells armed with the red bullets. So what are the functions of the eosinophils? So they destroy the parasites, especially helminths. So they attack the large parasites that are too big for neutrophils or macrophages to eat. And the second function is allergies and asthma. So they play a major role in asthma attacks, allergic rhinitis, eczema, and they release the chemicals that contribute to the inflammation during all the allergies. So the next function is it also modulates the histamine effects. So they help to balance or reduce the action of histamine released by the mast cells and basophils. So this prevents excessive allergic reactions. And the fourth function is phagocytose antigen antibody complexes. So eosinophils, they help in cleaning the immune system by removing the complex formed after the antigen antibody reactions. And here is a picture of eosinophil. It has bilobe nucleus which is shown with two lobes and red or orange granules. These are the large colorful granules filling the cytoplasm, and it has lipid bodies. So these are the storage structures which are visible inside, and it also has this Golgi apparatus which produces and packs the granule proteins, and it has this primary granule, and this contain MBP, and next is sombrebro vesicles. These are the small vesicle-like structure which are unique to eosinophils. So everything inside supports the parasite killing and allergy response. So the third type of immune cell is basophil. So basophils, these are the least common type of white blood cells, but these are crucial in allergic reactions and inflammations. So what is the structure of basophil? It has bilobe nucleus and it has large blue or purple granules. So bilobe nucleus, so it is often S-shaped, but usually it is upskirt, nothing but covered by granules. So this has large blue or purple granules. So the cytoplasm contains big dark blue or purple granules. So these granules are stained with basic dyes. So this contain histamine, heparin, and inflammatory mediators. So basophils look like the purple berries because granules cover the entire cell. So here is a picture of the basophil on the right side. You can see the S shape, right? So the nucleus resembles somewhat like a S shape. So it is usually hidden behind the dense granules. So it has large coarse granules. The granules are blue or purple, which is containing histamine, heparin, and inflammatory mediators. So it has glycogen, which is the energy which stores for rapid activation, and Golgi apparatus, it helps to produce the granules, whereas mitochondria, it provides the energy. So what are the functions of the basophils? So basophils, they generally releases the histamine, like they causes the swelling, redness, itching during allergic reactions. It also releases the heparin. It is a natural anticoagulant which prevents blood clotting. And basophils, they play a key role in allergy and anaphylaxis. So basophils, they are activated when allergies bind to IgE. So hemoglobin E antibodies on the surface. This leads to vasodilation, smooth muscle contraction, and airway tightening in the asthma. So basophils, they also work with mast cells. So basophils are like the blood version of mast cells which are found in the tissues. So both are central in allergic reactions. So next is mast cell. So mast cells, these are the special immune cells which are found mainly in the tissues, especially where the body interacts with the outside world like skin, lungs, gut, and nose. So they play a key role in the allergic reactions and protection against the parasites. So now what is the structure of the mast cell? So this has large tissue resident cells, like they do not circulate like basophils, they stay in tissues and wait for antigens. So they are packed with granules. So these granules, they contain the histamine, like they causes itching, redness, and swelling. They contain heparin. It works as an anticoagulant. They also contain the other mediators like serotonin, proteases, and cytokines. So these mast cells, they have the surface IgE receptors. So this is the most important feature of the mast cell. So this mast cell, they have high affinity IgE receptors on their surface. So IgE antibodies, this bind to these receptors and arm the mast cell. So this is the structure of the mast cell. So what are the functions of the mast cell? So immediate hypersensitivity type 1. So this includes allergic rhinitis like sneezing or itching. It also includes asthma, food allergies, hives and anaphylaxis. So what happens during an allergic reaction? So allergen, it enters the body like pollen, dust, food and antigen. So allergen, it binds to IgE antibodies on the mast cell surface. So what happens? Then this triggers the degranulation, so which includes the release of the chemicals. And next, effects of the released mediators. Histamine, it gives the vasodilation like redness, heat and swelling. So there will be an increased permeability. Symptoms like runny nose and water eyes. So also the other effect is bronchoconstriction. So this is seen in asthma, which they experience a difficulty in breathing, and also the increased mucus production like protective, but it causes the congestion, and also protection against the parasites. So mast cells, they help to expel worms through increased mucus and muscle contraction. You can look in this picture the mast cell. So here the IgE antibodies on the surface, you can see them. So these are the Y-shaped structures which bind allergens. So they are attached to the Fc receptors on the mast cell membrane. So next, antigen binding. So the green shape labeled antigen, it connects to the IgE antibody. So this cross-linking is what triggers the mast cell activation. And it has the histamine granules. So inside the cell, there are pink or purple dots which represent the histamine-filled granules. So when activated, these granules fuse with the membrane and releases the contents, nothing but the process called as degranulation. And there are complement receptors like C3A and C5A. So these are anaphylatoxins. So they can also activate the mast cells without IgE antibody. So next, degranulation. So what do you mean by degranulation? At the bottom, you can see the granules being released into the surrounding tissue through inflammation, swelling and redness. So what are the mediators released by the mast cell? Histamine, heparin, serotonin, leukotrienes, prostaglandins and cytokines. So this causes the prolonged allergic symptoms. So next, coming to monocytes or macrophages. So monocytes or macrophages, these are the part of innate immune system. So the main job is to eat and destroy germs, dead cells and foreign particles. So they are the best phagocytes in the body. So what is the structure and the basics of monocytes? It is the largest WBC. So they are the bigger than neutrophils, lymphocytes, basophils as well as eosinophils. So it has kidney-shaped nucleus. This is the easiest way to identify monocytes under a microscope. They also circulate in the blood. So monocytes, they move through the bloodstream until they reach the tissues. So monocytes become macrophages after entering the tissues. So second one is macrophages. What is the structure? It has a large granular cells and tissues. After monocytes enter any tissue, they transform into macrophages and they grow bigger. And it also produces lots of lysosomes. So because macrophages, they digest many materials, they have many lysosomes containing enzymes. So they are long-lived, like they can live for months to years. And it has tissue-specific names of macrophages. So different tissues have their own specialized macrophages, right? Like liver, the macrophage name is Kupper cells. So this filter blood commonly in the gut. And in brain, the macrophage name are microglia cells. So this protect the neurons. And in lungs, alveolar macrophages, so they remove the dust and particles. So in both, the macrophages are osteoclast. So what does they do? They break down the bone remodeling. So the next is skin, histiocytes, where phagocytose detects the pathogens. So all these are just different versions of macrophages which are adapted to their own tissue. And what are the functions of the monocytes and macrophages? So phagocytosis. So this is the main and best function of the macrophage and monocyte. They engulf the bacteria, dead cells, dust particles, cell debris and foreign substances. So the next function is antigen presentation. So after digesting the microbes, macrophages, they present antigens to the T cells. So this activates the adaptive immunity. And next, it releases the cytokines. Like they produces the inflammatory mediators like IL-1 and TNF alpha to alert the other immune cells. So they also play a role in tissue repair, like they help in healing by removing dead cells and stimulating the growth factors. And the previous picture, there is a monocyte and macrophage. So monocyte, it contains the nucleus, which is large, pale and kidney shaped, and it also consists of phagosome, which is a bubble-like structure, and lysosome, it is small enzyme-filled structures. Whereas in macrophages, it has pseudopodia, like these are the arm-like extensions that reaches out to catch the particles or microbes. So it has phagosome, which is a vacuum-like vesicle containing the swallowed bacteria, and it has lysosome. This contains powerful digestive enzymes, and phagolysosome. So when phagosome plus lysosome fuse, digestion begins. So this is where the microbe is skin line broken down, and this is why it is called as phagolysosome. So next, coming to dendritic cells. So these are the most potent antigen presenting cells. So these are the best antigen presenting cells in the immune system. So dendritic cells, they act as a bridge between innate and adaptive immunity. So what is the structure of the dendritic cell? These are the star-shaped cell, like they have long branching projections which are called as dendrites. So this helps them to reach out and capture the antigens. So now what is the location of the dendritic cells? This is present in skin, which is called as Langerhans cells. They are located in the mucosa like respiratory, GI and GU, and they are also present in the lymph nodes. So why this shape? Because these long projections increases the surface area. So this helps them to interact with the T cells efficiently. So now what are the functions of the dendritic cells? I already said you these are the most efficient antigen presenting cell. So they process and present the antigens to MHC2 to T helper cells, CD4 plus cells. So they capture, they migrate and they activate. So what are the steps? They capture the antigen in the tissues, they travel to the lymph node, they present antigens to the T cells, and at last they activates the adaptive immunity. So they act as a bridge between innate and adaptive immunity. They sense the pathogens, where innate pathogens, and they also activate the T cells. This is the adaptive process. So without dendritic cells, T cells always remain inactive. And the right side, you can see the picture of the dendritic cell. Left side, you have an immature dendritic cell. These are found in tissues. The main job is to capture the antigens. So this has fewer projections. And this immature dendritic cells, it undergoes maturation. It is triggered by pathogens and cytokines like TNF alpha and interleukins 1. And then it forms into a mature dendritic cell. So this mature dendritic cell, it is located in the lymph nodes. It has more branching arms. And the main job of this dendritic cell is to present the antigen to the T cells. So it shows the MHC molecules and the core stimulatory markers, which are not labeled but they are implied. So next is natural killer cells. So NK cells, these are a part of innate immunity, but they originate from the lymphoid lineage, same as the T and B cells. So they act very fast, within minutes to hours. So what is the structure of the NK cells? This has the large granular lymphocytes. So they contain the cytotoxic granules filled with the perforin, which creates spores, and they also has the granzymes, which induces the apoptosis in the target cells. So the NK cells do not have a no antigen specific receptor. This means there is no RCR or TCR. So they do not require the antigen presentation. So they kill based upon the balance of the signals. Now, what are the functions of the NK cells? So they kill the virus infected cells. Nothing but the NK cells target the cells that have low MHC1, a sign of viral infection. So it kills the tumor cells. If a cell behaves abnormally, NK cell destroys it. And it also produces the interferons beta. So it helps to activate the macrophages and enhances the immune responses. Also, it plays a major role as the ADCC. So what do you mean by ADCC? Antibody dependent cellular cytotoxicity. So this NK cells, they uses a CD16 receptors to kill the antibody coated cells. So NK cells, they respond quickly. There is no need for prior exposure and there is no memory. So in the previous slide, you can see the diagram of the NK cell. So it has the activating receptors. So what are the activating receptors? This turn on the killing function. So it has NKG2D, NKP46, NKP44, NKP30 and CD16, which is important for ADCC. So if activating signals, it increases the inhibitory signals which promote the NK cells to kill the target. And this has inhibitory receptors on the right side. So this inhibitory receptors, they stop the NK cell from killing. So what are the examples? KIRS and NKG2A or CD94. So this receptors, they recognize MHC1 on normal cells. So if a cell shows normal MHC1, the NK cell, it does not kill it. So it also has the core activating receptors NKP80, DNAM1 and 2B4. So this enhances the activation when combined with other signals. So this is about the NK cells. So next is T cells. So what are T cells? They are the central players of the adaptive immunity. So they develop in the thymus and help to coordinate, control and kill infected cells. So what is the structure of the T cell? These are the small cells. So these are more smaller than the most of the WBC. It has high nucleus to cytoplasm ratio. Like it has large round nucleus. It has a very little cytoplasm. So this is why they look dense under the microscope. So what are the markers of the T cells? So CD3, CD4 and CD8. So CD3, they present on all the T cells. Like this is the T cell receptor signaling complex. And CD4 marker, these are found on the helper T cells. They help other immune cells. Whereas CD8, they are found on the cytotoxic T cells. So they kill infected or tumor cells. So now what are the types and functions of T cells? The first type is CD4 plus T helper cells. So these T helper cells, they coordinate the immune response by releasing the cytokines that activate the other cells. So they activate B cells, which helps in antibody production. So they activate the macrophages, they activate CD8 T cells, they regulate the inflammation. So this CD4 T cells, they differentiate into subtypes again. Th1 cells. So they fight. What is the function of Th1? They fight the intracellular microbes. For example, cytokines, interferons, gamma. So Th2 cells, they fight parasites and drive the allergies. So these are associated with the IgE, eosinophils and mast cells. So Th17, what is the function of Th17? They fight against the extracellular bacteria and fungi. For example, interleukin 17 and TFH, nothing but follicular T helper T cells. So the main function of TFH is they help the B cells in germinal centers. Like they create high affinity antibodies, they promote the class switching. So here is a picture. You can see three types of T cells: the cytotoxic T cells, helper T cells and regulated T cells. So cytotoxic T cell, this has a blue body, so holding a weapon, nothing but it is symbolizing the killing function. So what is the function? Produces toxic agents to kill the targets. So it represents the perforin and granzymes. Next is helper T cells. So these are light blue in color, holding a green go sign. So what is the function? It stimulates the B cells to make the antibodies. It stimulates the T cells to become active. So this shows that helper T cells, they activate other immune cells. And third type is regulatory T cells. So these are pink in color, holding a red stop sign over here. So what does it mean? It suppresses the immune responses. Like it shows their role in controlling overactive immunity. And this is the T helper cells. So next is CD8 plus cytotoxic T cells. So what are CD8 plus T cells? These are called as cytotoxic T lymphocytes. So they are the body's killer cells of the adaptive immune system. So what are the main functions? It kills the virus infected cells. Like they identify the cells harboring viruses and they destroy them before new viruses are released. It kills the tumor cells as well. They recognize the abnormal proteins which are present on the cancer cells and they also induces the apoptosis process. What do you mean by apoptosis? It is a programmed cell death. So it also releases the cytotoxic molecules like perforin and granzymes. So what does perforin do? It forms the pores in the target cell membrane, and granzymes, it enter through this perforin made pores and it trigger the apoptosis, the programmed cell death. So this is the function of the cytotoxic T cells. So next, regulated T cells, which is called as Tregs. So Tregs are the peacekeepers of the immune system. So what are the main functions of this regulated T cells? It prevents autoimmune diseases, like it suppresses the overactive immune responses, and it prevents the immune system from attacking their body's own tissues. Like it maintains the tolerance. They teach the immune system to tolerate the self antigens as well as the harmless substances. So these are the important markers of the Tregs. CD4, CD25. So FOXP3, it is a master transcription factor which is required for Treg development as well as function. So without FOXP3, there will be no severe autoimmune disease. And here is a diagram which shows you the development and activation and differentiation of this T cells. So this section explains how a naive T cell develops. So the first is hematopoietic stem cell, HSC. So this is a small cell labeled CD34. So it represents an early stem cell in the bone marrow. So the stem cells, they can differentiate into T cells. So next is early T cell precursor. So as the cell progresses in the thymus, it expresses certain markers like CD2, CD5, CD7, CD3, and alpha gamma TCR. So these are the early T cell precursors. So then it differentiates into the T cell subtypes. Two main branches appear here. CD4 plus, which are T helper cells light blue in color, and CD8 plus cytotoxic T cells, which are dark blue in color. So the second is activation and differentiation. So this shows what happens when a naive CD8 plus T cell encounters an antigen. So first the pathogen triggers the immune reaction, then the immature dendritic cells that transfer into the mature, it becomes into a mature dendritic cell, displaying the antigen on its surface. And then after mature dendritic cell, it naives a CD8 plus T cell activation. So this activation requires the TCR binding to antigen and also co-stimulation. So next, clonal proliferation takes place and the effector CD8 plus T cell functions, and then it helps in killing the target cell. So this is how the development and activation and differentiation takes place. So this is for today's session. I hope this session has helped you. So in the next session, you will be learning about the types of immunity and as well as the physiology of immune system. Thank you.